All right, well, welcome everybody. Let me see, I'm gonna have to do this twice here. This is gonna be chapter 3, biology of behavior. Biology and behavior, I guess. So let me go ahead and share my screen. I think it's easier sometimes if I just have the screen, that way I can switch it around if necessary. So, all right, so this is in one of the lectures where I have extra things from the PowerPoint that are left at the textbook actually excluded. So it's good that you're watching this. So here's some examples. This again comes from a previous edition, but I think it's worth your while. Go ahead and tune into these YouTubes if you want to. It's not obviously optional. And this basically is kind of a preview. The basal ganglia, the thalamus and the cerebellum are all part of motor systems in the brain. In Huntington's disease, those are abnormal. Huntington's disease tends to have chorea. They tend to have a hard time moving, or a hard time not moving, rather. That's actually more the case. And it's an autosomal dominant disease, and it can be lethal. So that's just an example. So neurons receive, integrate, and transmit information. We have about 1 billion neurons. That's a lot, and 10,000 connections each. So they have neural networks. This is kind of the idea to think about neural networks. And these are actual pictures of neurons. There's a certain technique you can do that. So it's kind of cool. And this is actually a picture of a neuron in particular. It really doesn't do it justice, though, right? I mean, you can't tell that there's 10,000 connections there. So it doesn't really give it justice, but there is. That's a lot of connections. So kind of a beginning here, there's the central nervous system, which is basically everything inside bones, so the brain and spinal cord, and then the peripheral, which is everything else. We're generally going to focus on the brain, of course, but the other ones come into play as well. And here's a nice little listing. This is a figure 3.1 from your textbook. Central nervous system, brain and spinal cord, talked about that. The peripheral nervous system has somatic. This is skin, muscles, joints, etc. And send signals to the spinal cord and brain. So that's all sensory receptors, right? And then here, the autonomic is glands, and sympathetic and parasympathetic nervous system. So this is fight or flight, and this is I just had a big meal and I want to kind of kick back. Here's more. This is again one of the things that I kept in, because they have sensory neurons. They're afferent. They go at the brain, afferent. And efferent, the skin and muscle, other somatosensory neurons, etc. And then there's motor neurons that exit the brain, efferent. They exit the brain and go to muscles. And interneurons, which is the ones I want to talk about the most, are going to be the ones between the afferent and the efferent. Let's see. This is sort of interesting. This is this poor person touched a little tack there. Ouch! Moved his or her hand, and from the reflex, just goes to the spinal cord. Now, interestingly, that movement happens before you sense the pain, right? Because it takes a while for that to get up to the brain. And I think I'm going to use my pen here. I think that works better. It takes a while for that to get up to the brain. There's an eye. But so you have an activity that happens even before you sense it. Now, neuronal structure, dendrites, cell body, axon, terminal buttons, synapse. At the terminal axon, synaptic cleft, lots of words. Let's look at the next picture. And this remained the same from the 4th edition to the 6th. So there's no real reason to change it. Yeah, this is 6th edition. It's interesting how they change things, but they don't really change it. So anyway, there's the dendrites. They received information from other neurons, as you can see down over here. So basically it gets in here to the cell body. That's the nucleus. Neurons either fire or they don't fire. So here it decides to fire, say, goes down in the direction of impulse. Myelin sheath, and then from the oligodendrocytes is what they're called. Nodes of Ranvier. So the impulse doesn't have to just go straight down. It can jump from one to the other. We'll talk about that a little bit. And here's got the end of the axon, goes across to the synaptic cleft. So that seems pretty simple, one neuron to another. You've got to remember there's around 100 billion of them, and they have 10,000 connections, and they fire about 10 to 100 times a second. So quite the complex little thing we got going on in our skulls. Again, this is more of some pictures I kept around. This is kind of a nice little picture. Let's see. I have a new Apple Watch, and it's weird. it buzzes me every now and again. But this is an actual picture. So we're looking at this section here. Okay, we're going to dial down onto one of those. These little vacuoles, okay, those little things are filled with neurotransmitter. And through a stupid complex system, they get released, and they will cross that little cleft there and hit the dendrite and tell that the next neuron either to fire or not fire. Okay, it's excitatory or inhibitory. Those are words you get to know. Excitatory or inhibitory. So let's get a little bit more of this. Let me make sure I get my timing correct. So myelin sheath, nodes of Ranvier, ion channels at the nodes. This is ions are like sodium, right? Sodium chloride, those are ions, and potassium, those are ions. They're ionized. Then gray matter has the neuronal cell bodies, and the white matter is the myelinated axon. So this here, okay, the myelinated axons. White. The myelin is essentially fat. It's a phospholipid bilayers or lipid. So fat, like bacon, if you like bacon, is white. So the white matter is made up of fat. It's the thing to think about. Now, glia I kept in. Then this is they're pretty important. So I want you to keep these in mind. So astroglia, they surround those synapses. These little astrocytes, they surround the synapses, and they coordinate the firing. And like one can get up to like 1 million of them, right? It's just kind of interesting. And they make, of course, there's 100 billion neurons, so 1 million isn't really that much. So, but that makes it kind of cool because it makes the neurons sort of fire synchronistically, coordinate that firing. The oligodendrocytes, we talked about those already. There we are, kind of wrapping around the axon. And the microglia are basically white blood cells that come from the bone marrow, and they get into the brain, and they have to do with fighting infections. So if you get like a viral infection in your brain, it takes care of that. Pruning, we have a lot of different neuronal connections that we don't really use when we're just a kid, so they prune those off. They may be involved, and actually it's fairly good data that they're involved in like schizophrenia and brain injury, that they may have abnormal functioning with schizophrenia and brain injury. So pretty important. Microglia are pretty important cells. The glia in general are pretty important cells. They're really as important as neurons, really, because and they too. There's actually more astroglia than there are neurons, or excuse me, glia in general than there are neurons. You may have heard of brain tumors. You've probably heard people get brain tumors. Astrocytes, the astroglia, gets is one of the main types of brain cancer. Astrocytoma, that's what they're called, astrocytoma. Here's the membrane potential. This is a figure 3.4, page 73 in your textbook. And so they're polarized. What that means is there's more positive ions on the outside, sodium on the outside, than there are the negative ones on the inside. So that's chloride, actually. Sodium chloride. So sodium chloride is like salt, right? Table salt, sodium chloride. Potassium chloride is another kind of salt, but table salt is sodium chloride. So because they have the more positive on the outside than on the inside, that's called polarized. Now, if you have a magnet, basically those want to go together, right? And they also just diffuse. So if there's more in one place, will diffuse to the other side. So that's what they have. That's what a neuron firing is. That's what a neuron firing is. So when they fire, those channels open up. Okay, those channels open up, and ions get passed through. Now, in order to repolarize them, they depolarize them, pass through. You have to repolarize them for the next one. They have these pumps. Sodium, potassium pumps. They bring the sodium back in and put the potassium back out. So those are the idea. sodium channels. Now we'll be getting introduced to those quite a bit, and we get a different color here because I just want a different color. These ion channels, they're sodium channels. Those become very important when we talk about bipolar disorder, seizures, that kind of stuff, because they basically a lot of the medications that for bipolar disorder are sodium channel blockers. Okay, so when someone's manic, their neurons are firing too quickly. So if you block down the sodium channel, it slows them down, and all things go well. The maniac is treated. So here's the neuron firing. Here, this is the idea. This is actually on page 74. It's basically here's polarized, right? Here's polarized. You have to have this threshold of excitation. Okay, threshold of excitation. The neuron, it has to have enough impulses to hit that threshold. Once it does, it fires. Okay, once it does, it fires. There's nothing to stop it. It fires. So it's all or nothing thing. All or nothing thing. Let me see. So let me erase some of this because I want to go back here. There you are. So erase. There we go. Now back to get a pin. So this is important. So if you let's see, I don't think I have this yet. Let's go back here. So this is where a lot of medications and drugs use. So I wish I can make this bigger. That's okay. Basically, if you're using methamphetamine, say, this is lessened. So let's just say it goes down to there, or up to there rather. That means you don't need as much stimuli for it to fire. Okay, that's called you're buzzed on methamphetamine. Okay, so that's what that does. Okay, it reduces it. Let's say you have, well, we'll have alcohol, we'll have red wine. That actually increases it. Okay, so the space there, the depolarization is now increased, which means it takes more stimuli in order for it to fire. So you have a few drinks, you feel kind of relaxed. A bunch of drinks, you pass out. So that's why. So reduce or increase polarization. So meth and alcohol. There you go. And lots of other things do that too, obviously. This is going to be that little increasing or decreasing the polarization has a lot to do with everything else we're going to talk about as far as this chapter and really the rest of the text. Of course, I don't know if this is going to work. No, it's not. Yeah, I should just get rid of that. It's a cool little picture of it, but obviously it's not working. Sorry. So here's the all or nothing principle. The potentials go along the axons. The entire process takes about 1,000th of a second. So that's pretty damn fast, right? That's a lot of activity. So it's really pretty amazing, right? When you think about all this, there's 100 billion neurons, 10,000 connections each, firing all the time across your brain. It's just really quite an amazing thing. We already talked about the all or nothing principle. And here's the neurotransmitters. Okay, so they have various neurotransmitters that we'll cover in a second. So this is, again, we're sort of dialing down here to there. So there's neurotransmitters being made. They put in those vesicles that we talked about earlier. They get the impulse comes down. Wow, it's time to spit out some neurotransmitters. They spit it out. They go and they hit the receptor on the other side. Okay, the receptor, the target receptor, and they're either broken down, taken back up, or this basically says, whoa, there's enough neurotransmitter in the synapse, and we don't need any more, slow her down. So it goes down to the axon terminal, or terminal button. They have a bunch of different names for it. The synaptic cleft, space in between, and then the dendrite receiving the signal with these different receptors. And there's several different types of receptors, by the way. So this is where a lot of cool stuff happens as well. I'll get my eraser. I like my little eraser. So you can block, so this will use depression, okay? You can use a monoamine oxidase inhibitor. So these are monoamines. Neurotransmitters are, many of them, not all, monoamine oxidase. Okay, they're monoamine oxidase. They basically oxidize the neurotransmitters. So if you inhibit them, monoamine oxidase inhibitor, maybe I'll link, make a note to myself here. I'll link the MAOI. You'll like this. make a note. So I'll do this. I'll link it for you to watch. It's pretty funny. It's MAOI. If you inhibit that, it treats depression. Or you may have heard of things like Prozac or Zoloft and whatnot. Those are serotonin reuptake inhibitors, so they block that. All of those things give you more neurotransmitter in the cleft. And however that works, we don't quite know, frankly, but that corresponds to having your depression improve. Now, you can have agonists that act like it. The agonists, they act like the neurotransmitter. And moving along here, this is figure 3.7, act like the neurotransmitter. They can, by doing that, they can block the reuptake. We talked about that. They can also block the monoamine oxidase, so that's, they don't really show that here, but they can. Or they can mimic it on the receptor, on the other side, over here. Antagonists, the main way antagonists work is they block, okay? They block this. So you can have sodium channel blockers, because neurotransmitters sometimes will just act straight on the sodium channel. You can have just receptor blockers. For example, with someone with schizophrenia, they take an antipsychotic medication, it blocks the dopamine receptors. We'll get to all of that in a bit. That block the dopamine receptors, and that treats the schizophrenia, assuming everything goes well. And you also have partial agonists. What that means is they when there's a lot of a neurotransmitter, they block it. When there's not enough, it's a matter of affinity. Blocking with affinity, how much it sticks to it is basically what that is. And here's a list of various ones. Table 3.1. This is a new one. So there's, again, a bunch of them. Acetylcholine, norepinephrine, serotonin, dopamine. There's the ones listed. There's a bunch more, including anandamide. That's the endogenous cannabinoid. So that, again, we have endogenous opiates. These are opiates, actually. It's actually opioid. Let me do that again. Eraser. Okay. They're called opioids. Opioid receptor. So when you think about it, of course, we have things that the opium acts on and heroin acts on, opiates, because if they didn't have something to receive it, you wouldn't have any effect from the opiates. So that's the opioid receptor. And similar, this is the marijuana receptor. This is cannabinoid receptor. There's a bunch of others, and medications and/or drugs that work on each of these. And we'll continue to cover those as we go through the course. So this is the big idea, right? You have these bunch of neurons, axonal terminals with ion channels, various neurotransmitters. You can increase them or decrease them, their activity, depending on what you do. That's the whole idea. Okay, that's the whole idea. There's a lot of details in there, but once you get that sort of big picture, that's really, you'll be in good shape after that, really. Now here's some slides that I just want you to get a good impression of. Okay, and I show these all the time. I love these slides. So norepinephrine, these are long axons here. Okay, this like those little neurons. There's a bunch of them here, and their axons loop around the brain. They stem. They kind of move around as they have these branches as they move. So now you're kind of getting a feel for the 10,000 connections. Okay, instead of the little cartoon neurons, these are now neurons that loop around across the brain. So that's a lot. But when they do things, here's all the different things that they do. They're involved in depression, attention. So ADHD, for example, agitation. They have impulse dysregulation. See, so up through here. So all this stuff is part of the norepinephrine. It's also involved in negative emotions. Okay, so anger, that kind of stuff, etc. So again, I want you to have a big picture of these things. Serotonin, you probably know that one quite a bit. So it goes to the frontal cortex for mood regulation, impulse control, actually through here. Other things, anxiety, hypothalamus for eating, etc. Sexual response to the spinal cord, vomiting, all these things serotonin does. And another thing that I want you to keep in mind with these is that medications have side effects, right? Medications have side effects. They all do. Of course they do. And that's why, because they don't just do one thing. Serotonin doesn't just do one thing. It does a bunch of different stuff. Okay? It does a bunch of different stuff. And therefore, medication will act on all serotonin receptors. No, a lot of them actually. There's a bunch of serotonin receptors, so I'll just say a lot of them. So that's the big picture of what I want you to get here. Dopamine, 4 dopamine pathways: substantia nigra, mesolimbic, mesocortical, and hypothalamic pituitary. You know, these are very important as well. We'll talk about the 4 pathways a bunch of times. So the nigrostriatal pathway goes from the substantia nigra to the strial. Remember that basal ganglia from way back early on? No, let me go back. Hang on. Boom, boom, boom. Oh, wrong way. Go back. Here we go. Basal ganglia. Okay, that part of the brain is this part of the brain. They're looking, they're looking different directions, but that's the same part of the brain. That's involved in movement. So decreased activity from the dopamine there, you get Parkinson's-like disease. Okay, mesolimbic. Okay, goes from the meso means mid, like mezzanine. People don't know what mezzanine is generally, but it's like the mid layer between the lobby and the first floor of a hotel. It's mezzanine. But it goes from mesolimbic is pleasure, hallucinations, and delusions. So people who have schizophrenia have too much activity there, for example, they have hallucinations. And then mesocortical, that's executive functioning, planning things out, deciding what you want to do. Higher cortical things, making decisions. And finally, the hypothalamus to the pituitary, okay, that blocks prolactin release. It's blocking it. So let's say you have someone with schizophrenia, and you need to give them a medication to block the dopamine in the mesolimbic system. And so this, where do we go here? Mesolimbic. Okay, cool. That decreases hallucinations. Medications like haloperidol do that. They decrease hallucinations quite well. But they also have movement problems. They'll get Parkinson-like disease, right? It's called Parkinsonian. It's not Parkinson's, but it's Parkinsonian. It's like Parkinson's. Okay, so they get stiff. They also will block the pituitary and increase prolactin levels, and that will have people start producing breast milk. So prolactin is like lactose, right? Lactation with breast milk. So if you block prolactin, like golactin or prolactin, so if you block it, you don't get breast milk. If you block the blocker, prolactin is released. People grow breast tissue, including boys. There's actually a class action lawsuit with Risperdal for adolescent boys who got breasts by taking Risperdal. The choline, again, sort of starts here and works its way back. Better have a different color because that one's red. Haven't done blue yet. Work its way back. This is involved in Alzheimer's. Okay. The acetylcholine is one that's decreased in Alzheimer's disorder. Memory, learning. Decrease those things. It's called Alzheimer's. It's also at the neuromuscular junction. That when the neurons hit the muscles, you block it. If you block that, you get paralyzed. People get Botox, right? This botulism toxin, by the way, botulism toxin, it kills people. But they squirt a little bit in their forehead, and they no longer can wrinkle their forehead, and that's supposedly looks better. I look stupid myself, but what do I know? GABA, gamma amino benzoic acid, GABA. Everybody calls it GABA. It's inhibitory. Okay, it's inhibitory. It slows down the next neuron. Yeah, that's it tells the next neuron to not fire. So going back to here, I'm going to try to draw all these things together. It increases that. Okay, increases the resting potential. That's how much polarized it is, and makes it harder to fire. I want to kind of get the big picture for you guys and gals. So it makes it harder to fire. So if you have anxiety, a little benzodiazepine might do you some good. And if you're having some kind of medical procedure, a little bit of benzodiazepine, you won't remember it. That sort of idea. MDA, glutamate, it's excitatory. Okay, it's excitatory one. Medications can act on that as well. Okay, so it's excitatory. So that one makes you go up, lessens that space between the resting potential and the threshold of excitation. Goes up. A little neuron fire in there. Kind of dropped off at the end though. Back there. So basic brain structures. There's some just basic ones here. Broca's area, history. This is Broca's area. Basically, people would get strokes, and they, with the strokes, they would not be able to talk. Broca, that's the guy, would do the autopsy on people who had strokes and found out, oh, that's the part of the brain that was dead from the strokes, and therefore that's where where speech is. And sure enough, it is. It's not the only place where speech is, but it is a big part. And this is the left hemisphere too, by the way. Phrenology is bumps. This is a figure 3.12 on in your text, page 81. Excuse me. The idea was that these little bumps on your head would have your personality. It's not true. We already saw these. I just think they're cool. By the way, with TMS, here's the Broca's area here. And if you can stop that area with these transcranial magnetic stimulation, you just get magnet and kind of you take away and you talk again. You take away and you talk again. Wouldn't that be handy in certain situations? Shut up. But no, we don't have it that way yet. We're working on it. And some more imaging. Let's see. Anything else I want to show you with this? Again, just really putting them in there in the machine and seeing all the different, all the different activity. It's pretty cool, huh? How detailed they get. And again, here's the structure. That's functional magnetic imaging. So they see usually it's the red's more active and blue's less active. So whatever they're doing, whatever they're doing in there, thinking, in there being the thinking, well, we can see it on the imaging. It's pretty cool, actually. There's some studies where they looked at people dreaming. They had them in these things while they fell asleep, which is quite something because they're quite loud, but they did. They fell asleep when they started to dream, and they get the rapid eye movements. And when they did that, they woke them up and told them were told what they were dreaming. And after a while, they could tell what they were dreaming by looking at the brain patterns. That's pretty cool, huh? We can read your dreams. Scary cool, but cool nonetheless. And here's another one, transcranial magnetic stimulation. Again, this is used quite a bit now for in psychiatry for treating mental illnesses. So that's really pretty cool. Now we're going to work our way up. Some of these are just, excuse me, sorry about that. But sorry, this is a figure 3.2. The brainstem has medulla oblongata. Pons, midbrain, reticular formation, and this is straight up just anatomy and physiology, right? This is what you need to know, A&P. Okay, so sleep and arousal, medulla has a lot to do with the breathing. Midbrain, that's the midbrain is where a lot of those neurotransmitter systems started. Remember the ones that kind of go around? A lot of them start in the midbrain, and then they loop around through this gyrus here, actually, cingulate gyrus, and they branch off to different parts of the brain. Pons has a lot of cranial nerves. Cranial nerves are basically nerves that don't leave the cranium. Okay, so that's the term cranial nerves. And reticular formation wakes you up. You can see it there. So cerebellum, some more there. Movement it's more coordination of movement. Like if you reach out and grab something, I'll give myself something to grab here. So I have a ruler. So if I want to reach out and grab the ruler, my cerebellum's helping me do that. If I didn't, I wouldn't be able to catch it as well. And actually, we can test your cerebellum if you want to. Do this. Are you done? How many of you actually did that? If you did that well, your cerebellum's intact. If not, you can't do it with the cerebellum that's not functioning. But it has lots to do with other things besides procedural memory. We'll talk about that's memory on how to do stuff, like a procedure. Right? People have procedures, but it also plans, remembers episodic things, episodic episodes in your life. It's involved in language, emotion. So it has actually more neurons in the cortex, but it's really pretty interesting. But it has more neurons in the cortex. So they say motor function. It has a lot more than that. Whoops, go away. Moving right along. All these different things. Here's the limbic system, amygdala, hippocampus. Hippocampus has to do with, I need another color here. What color do I want? How about just black? Hippocampus is with memory. Okay, so we'll have time, some things when we get to the memory chapter, some videos on people who had their hippocampi damaged and they can't form new memories. It's pretty quite something. Hypothalamus regulates body function, yet to say the least. It regulates a bunch of it. Take biopsych. Some of you going into the BSW program. I don't know if anybody in this class is, but there's not very many of you. But if you do, you'll take biological psychology, and you'll learn about the hypothalamus. There's like a bunch of different nuclei in there. Nuclei being a bunch of neurons together, and they regulate everything. Like they regulate your hormones. Hunger, sexual drive, your kidney function, thyroid function, not so much kidney function, but a bunch of stuff. And then the amygdala is for very strong emotions, fear and anger in particular, but strong emotions. And then the nucleus accumbens, which your text doesn't have, but I do because I want you to learn it. Yeah, so it's dopamine, and it has a circuitry between nucleus accumbens, which is pleasure, okay, to the prefrontal cortex, which is you're again organizing, executive functioning, interpreting the world, and that goes down to the amygdala, which is those strong emotions. So this is a nice little neurocircuitry, okay, that whole system right there, and we can see that being involved in anxiety disorders, depressive disorders, all that sort of stuff. So those are that's a good neurocircuitry for you to keep in mind. Let's see, there's a bunch of stuff there. Let's go to the next picture. So frontal lobe, temporal lobe. Let's see. There's various ones. We'll cover that YouTube in a little bit. But parietal lobe is for touch, spatial relationships. It's all about space. Temporal lobe is about time and also has hearing and memory. The hippocampus. Okay, this little hippocampus is on the kind of inside. It's medial on the inside of the temporal lobe. So hearing, memory, vision for the occipital cortex, and frontal lobes as thought, planning, and movement. Now we have some primary cortices here. These are important. So primary somatosensory, that's where your body comes up and touches it. Okay. Primary visual cortex, that's where vision first gets to. Yeah, primary means it's where it first gets to in the brain. Okay. Once it hits there, it expands out to various other areas, but it starts there. And primary auditory. Smell is like here, okay? It's sort of in there. Here's the corpus callosum. You want to call someone, you want to call somebody, corpus callosum, you call somebody, you call the other side. So it's connected to a huge connection there. Now this is figure 3.24, the corpus callosum. Let's see. There's millions and millions of axons going through there, but we do basically have two different brains. It's kind of interesting that we have this two different brains that they have very different functions. Here's a nice little homunculi. So this is the motor one. Okay, motor cortex. It's on the frontal lobe, so motor and sensory on this side. So yeah, you probably have heard of like phantom hands. Okay, phantom hands. So what happens is if you get your hand cut off for whatever reason, you lose a hand, your brain still expects it. Okay, better get another color. The brain is still expecting to feel it. Okay, but it's not there. So the brain will basically generate the feelings on its own. Okay, which is weird, right? Because you don't have a hand, but you feel your hand even though it's not there. So is this the? Yeah, so that's phantom pain. And you get phantom hearing, phantom vision actually. Let's go back to here. So there's a thing called macular degeneration, where the basically the back of the eye dies. So you'll start having basically phantom vision. You start having visual hallucinations because the eye is gone, but the brain's still there. So you get phantom, all kinds of stuff. So split brain. Let's see. I think the Gazzaniga changed this up a little bit. No, it's here. Here's a split brain. This is figure 3.29. Splitting the brain splits the mind. Gazzaniga was really into this. This is one of his main things, so he talks about this a lot. But they basically cut the corpus callosum. So here, whoop, cut, slice. Fair enough, they slice it, and now you have two brains. There's a cool video on that too. And we'll watch that. Here again, it's the same thing. So what happens here, and we'll cover this in a couple chapters from now, things on the right, your right visual field, go to the left cortex. Okay, things on the left visual field go to the right cortex. They cross over. So this part of the brain, Broca's area, is with speech. What happens is if you show people who have that corpus callosum split something in their left visual field, they won't be able to say that they saw it. They say they didn't see it, but they'll be able to draw it. And with the left hand, it might be a, yeah, it's really quite something. But how are you doing on time? We're doing good. And this shows that they can't see it. They can say it's a fork, but the other one sees a spoon, so they reach out and they grab the spoon, even though they can't report that they saw a spoon. Okay, they just go grab it. So the brain is functioning even though they can't talk, which is pretty interesting, right? It's kind of trippy. So it's kind of a neat thing here. Show that a fork, so they say a fork, but whether they grab, they actually grab the spoon. Okay. It's a cool experiment. Let's see. So peripheral nervous system, how are we doing here? Kind of covering this again. That's that slide again. They re-show it. So now we're down to this side, all the green. And here's the autonomic. So sympathetic is fight or flight. Fight or flight. So if something scares you, like, ah! Hopefully that scares you. If something scares you, probably you got a little bit of your heart race a little bit. You're going a little bit. Your eyes pupiled up. You got bigger a little bit. So that's the sympathetic nervous system, fight or flight. Sorry about that. But you have to demonstrate some of these things, right? Parasympathetic is the opposite. Your pupils constrict. Your bronchi constrict. Your heart rate slows, etc. Now, people who have asthma, okay, their bronchioles are constricted. That's what asthma is. So you can give them epinephrine. Okay, epinephrine is sympathomimetic. It's norepinephrine. It's the same sort of deal, but it's epi, kind of a little bit of a variation there. That then relaxes the bronchi and treats their asthma. That's how that works. And here's a bunch of glands. Okay, the various types of glands. Yeah, a picture over here. Yeah, this is now figure 3.36. 3.36. They have all these different ones, and they can see all these different things that it does. Pineal has melatonin. You've probably heard of melatonin. People take melatonin to help sleep. Pineal takes it, releases melatonin to help people sleep. A lot of people take melatonin exogenous, so they take a pill. And apparently it works pretty well. People sleep pretty well on it, and it doesn't look like it has long-term problems. It seems like it might, right? Because you're taking this external melatonin, but it seems like it doesn't actually. So thyroid controls how body burns energy. You probably heard of hyperthyroid. People get kind of wired and lose weight, and hypothyroid, where people are kind of sluggish and tired. So that's the thyroid gland. And you get calcium with parathyroid, thymus with the immune system, etc., etc. Adrenal. So adrenal is on top of the kidney glands. Adrenal glands. It's immune system, but it's also the fight or flight. Okay, there's also fight or flight. It also has to do with this system. Okay, it's not just sympathetic, but it's also the adrenal. There's a thing called HPA axis. Here we are. HPA axis, which basically is the hypothalamus, which is like in there, the pineal, the pituitary. And that then goes to the adrenal, so HPA. We'll be talking about that a little bit later on too. So HPA axis, that's involved in stress. Okay, it's involved in stress. So the hypothalamus tells the pituitary to tell the adrenal to release adrenaline. Actually, it's adrenaline. It's also called epinephrine. So release adrenaline. And there's a psychoneuroimmunology. I kept this one in. This is actually from Nature Reviews. Immunology is pretty cool. Basically, the hypothalamus controls pituitary, which gets down to actually controlling the immune system. T cells, B cells, NK cells, all these are part of the immune system. So it's kind of cool. So how does the brain change? The experience fine-tunes neuronal connections. So you got to have the experience when you're a kid in order to have neuronal connections. And there's always a thing female brains versus male brains. Out here, yeah, male brains, female brains. And sure, there are some differences, but let's do some. Welcome to science. We're now going back to the science. So you have these things called bell curves. Better have a purple. Okay. Bell curves. Remember the standard deviation, those lines? We have the bars. Had the standard deviation. Those are essentially the bell curves. Okay, bell curves on there. Let me erase that, it's getting confusing. So sure, we'll say the red's females and the purple's males for just arbitrary, no particular reason why. And you might say, you know, we're looking at this part versus that part. Better have a different color now. Go back to my eraser. What color do I want? How about green? So this area, that's the mean, right? That's the average. Sure, those are different from each other. We'll say p equals 0.05. You know, so yep, statistically significant. Sure, that's cool. Whatever. What are you going to do about this person? Is that a male or a female? Okay. So are they different? Sure. Are they that different? Not really. So it's a difference that makes no difference. That's the thing. Brains can recover from injury, which is cool. Okay. Let's see, they have a recover from injury. They had that picture already. What we need. They can. N-acetylcysteine, by the way. N-acetylcysteine is can really help your brain. NAC. It's an over-the-counter thing. Get it like the real food store or real grocery or a natural grocery, grocers, I guess it's called. And so that's, yeah, it's actually very good for brains. And it's over the counter, so I can recommend it. And they have a lot on the human growth hormone, chromosomes, phenotypes, etc., selective breeding. Well, actually, the Huntington's. Remember the Huntington's from the first couple of things, first couple of slides there. Huntington's is autosomal dominant. That means if you get it, you're going to have it. Okay, it's not the autosomes. It's not the XY chromosomes. It's the everything else. So if you have that, even though you don't need two of them, you just need one. So you could theoretically get rid of Huntington's disease in one generation by just having them not breed. No babies. It would actually go away, but you know, we're not doing that. So evolution, inheritance, variability, selectivity, get you evolution. They have mutation, all this sort of stuff that they talk about. They have flowers and stuff in your textbook. I don't think we need to do it. A bunch of stuff has genetic components. Pretty much everything does, right? Pretty much everything does. Your political affiliations, there's like genetic components to that. Religiosity, genetic components to that. Personality, yes. Various disorders, yes. But nothing's completely genetic. That's a very major point. Nothing is completely genetic. Yeah, so that's an important point. And so when we think of genes, we think of like determined. Oh my God, it's in my genes, right? Well, it's not. It's not true. It's not. It's just, yes, it's partly in your genes, but partly not in your genes. Part of your experience. So advantageous. We already have all that. So here's genetic expression can be modified. That's epigenetics. So this is an interesting one. MAO, monoamine oxidase, right? That's the thing in the middle there. Let's go back to that slide. Hang on. Here we go. Go to review. Too far. This one here, monoamine oxidase, MAOI. Review, remember all that, It's going to be cool. Cortex, I'm going to put it again. Too bad. Now, so if it has low activity and they have violent crime, and they have maltreatment as children, the low activity has more violent crimes. Higher activity of MAOI, even though it does go up, okay, it does go up with the maltreatment, it doesn't go up as far. Now, science time though, science time. This goes to 60. Okay, it should go to 100. If it went to 100, it wouldn't look as much, and it's still only 30. Okay? And what don't they have? Making sure I fulfill my promise from the last chapter. They do not have the error bars. You need to have the error bars, the confidence intervals, all that kind of stuff. If you don't have that, it really doesn't give you as much information. So you need those the CI, confidence intervals, or the error bars. This is a cool TED Talk. I don't know how to do these really with these classes. I just, look at it. Okay, look at it. It's really cool. Basically, what this does, the optogenetics, and they have a picture of it on page 116, figure 3.53. They basically put in these receptors that come from like algae into neurons, and they act like. Review again. To the good receptor here. They act like these. They act like these receptors with light. So they shine light into the brain, and they can open up these dang things. Okay, they can open up these receptors in the brain and change behavior. They can make the neurons fire or not fire by simply putting light into the brain. That's really, really amazingly cool. So kind of scary cool. So would you want that? Would you want to have this little tubes in your brain? Say you have seizures, right? So you have seizures to find out where it is. Start getting one, light comes on and stops it. Be kind of cool. Epigenetic again. Epigenetic, that's on top of the genes. Our behavior changes the way our DNA is expressed, and therefore the way the receptors are expressed, the way neurotransmitters go on to them express, all that sort of stuff. It's pretty amazing. So here's phantom limb. So in this person, see how the hand is close to the face? Well, they now are feeling their hand down here. Hand, very bad hand, but hand nonetheless. You can almost make that out. They feel it on their face. Think about that. I can feel my hand, but it's coming out of my face. It's pretty bizarre. It's pretty bizarre. This is what happens. Brains do that. Synesthesia. We don't talk about this as much, but if you can look at this and see a word, then you have synesthesia. Synesthesia is a mixing. I think they cover it this more in sensory, but that's okay. I like it here. So if you can see, you can kind of make it out, right? There's the 2s there and the 5s, but it looks kind of wavy. But if you see the word, you have synesthesia. Some people, the word, letters and numbers have color. So S's are red or whatever. I had one student actually see it in all my years of teaching, because the 5s are sort of dark brown and the 2s were light brown, so she could see it right away. And this is harder to do when I'm not on these things as opposed to the whiteboard. It's class. People always want me to write it out. Sort of. Yep, I missed it, didn't I? It spells color. Just trust me. Write it out on your own if you want to. Yeah, but it spells color. This is just too hard to do on this computer screen. Sorry. And male and female. Again, we talked about that. Males have larger brains. Women's are more connected. Let's see. This is all kind of left over from previous chapters, but ovulation changes behavior. When a woman ovulates, they're more attracted to muscular men. They receive better tips as a waitress. Okay, when they're ovulating, they're more likely to wear more revealing clothing. This is from the previous text editions, and they're not attracted to whiny, needy, nice guys. So it makes sense, right? The DNA wants to be shared there. The egg is being released, so they're more likely to have affairs at that time as well. All right, so I think that's it. Okay, I don't want to keep the annotations. Let me get away from here. Where are you, little Teams thing? Here you are. So, stop sharing. Okay, good. We're still recording. That's good. Let's see if it actually works this time. And again, nobody showed up here today, but that's okay. Nobody showed up. You don't have to. So I'll just I'll keep doing these. And goodbye. Stop recording.